Hello everybody, welcome on today's webinar about spray drying strategies. My name is Patrick Lange and I support Biki with leading online webinars. Our panelist today is Deborah Repko. She's application and technical support manager working for Biki since 2006 and supporting the spray drying and encapsulation business. Deborah has a tremendous and great background in development within the materials science industry. Deborah, I'll give the audience to you.
Good morning, everyone. Some of you are joining us from the West Coast in the United States. Others are from the far reaches of Europe, and I saw someone from Australia. So welcome, everybody. My objective today is just to get everybody a little more comfortable with formulation strategies in spray drying. A little bit about Biki before we get going into the full webinar. Biki is a global provider of laboratory equipment. Parent company is a Swiss company and is 81 years old. Biki Corporation is one of the many marketing organizations globally, and we are on the East Coast of the United States. Biki provides products and solutions to all industries, and we sell 11 different product sets. We are known as the leader in evaporation technology and the inventor of the rotary evaporator. We are also considered basically the leader in laboratory evaporators, which is what we're going to be talking about today. So what I would like to do is start with some basic principles of spray drying so that everybody is on an even playing field, not knowing what everybody's background is in spray drying or experience in that regard. Talk a little bit about the B290, but that's not really the emphasis of today's presentation. Going to hit upon current trends, talk about important finished attributes or product powder characteristics that spray drying can do for us. Going to explain the process parameters within full detail. Touch on encapsulation via spray drying, and then I'm going to hit different formulation strategies for different market segments. Okay, there will be overlap, I believe, in all of these. So even if you're not doing pharmaceutical and doing food, I think there's applicability. I'm going to touch on 5 different case studies or literature references. All of those references have been uploaded as handouts, which you can download for your further reference. In the end, we're going to have a live question and answer, and I will do my best to answer your questions. If there's something that's very in-depth or very specific, maybe we can handle that offline. Here's the principles of spray drying. Just a nice little depiction. So at the top of the spray dryer, we have our formulation and our atomizing nozzle. And we're going to have incoming compressed gas. On the back side of the dryer, we're going to cool ambient air over a heater. And we're going to heat this up high enough, which is called our inlet temperature, to effectively evaporate the formulation. We're going to atomize the formulation going into a drying cylinder. We're going to make our way through the system, creating an outlet temperature. That is the temperature that our product is being exposed to. Okay. If our product is nice and dry, we're going to see a vortex of material going through the cyclone, and then we want products in our collection vessel. We have a filter on the back side, so little smaller particles that can't be captured go to a filter. And on the back side, we have the aspirator that continues to pull ambient air through the system. So we'll call this a semi-continual process. So just to hit quickly on the B290, we would consider this a traditional laboratory dryer. It has a co-current flow, meaning in one direction. All of Bookie's equipment has glass so that everything is fully visual, which makes it nice from an R&D perspective. We can see what's going on. It has standard or traditional cyclone separators. From a throughput perspective, we can do up to 1 liter an hour. I caution that that's the maximum throughput based upon your percent solid. Okay, we want stuff to evaporate at the end of the day. Small sample, about 50 mils. Particle size, traditionally we would say 2 to 25 microns. Max inlet temperature is 220, which is very hot in my opinion. And residence time is quick, meaning how quickly does my wet droplet evaporate and make its way all the way through the system to the collection vessel is 1 to 2 seconds. That's pretty quick. Our spray dryer and many spray dryers can be configured to safely work with both aqueous systems and organic solvent systems based upon our formulation needs. So we have equipment to be able to safely handle the solvent. So I want to hit on some current trends. So I think the biggest application area really is what we call spray-dried amorphous solid dispersion for oral delivery, meaning I'm going to make a tablet or capsule from that. Controlled release products, once again, meaning from a formulation perspective, I'm going to pick a specific excipient to give me either controlled release, stained release, or quick release. I can do temperature-sensitive products, big molecules, vaccines, proteins, enzymes, functional food, alternate proteins, flavors, and natural products have always been a big mainstay of spray drying, and I think will continue to do so. Alternate battery technologies, nanomaterials and ceramics, and hemp and cannabis as well are sort of a new emerging market. So why would I want to do spray drying? What are the benefits and what can I do from basically a particle engineering perspective? First and foremost, I can design the particle size from my formulation. I can change the flowability, compressibility, and even bulk density of a given powder. I can do granulation and agglomeration of slurries. One really nice thing about spray drying is through my excipient or carrier choice, I can do taste masking. If I have poorly soluble drugs, I can increase both the viability and solubility, again, through excipient choice, and we'll hit on these through examples. Once again, quick residence time and a one-step encapsulation. So these are sort of the benefits, and through our examples, we'll look at a handful of these. So process parameters. I like to say this is sort of the meat and potatoes, so to speak, or your vegan and your vegetables of spray drying. So we have parameters that we have to set. At the end of the day, this is a processing tool, and I have to come up with proper settings. So we have to basically develop a process. So I can set my inlet temperature. I can adjust my feed rate, my atomized atomizing gas rate, my aspirator, that motor in the back, and I have complete control of my formulation. I'm going to get the resulting outlet temperature. I cannot set that on most laboratory dryers. That's an effect of the other parameters. How dry does my formulation get? What particle size I'm going to achieve, or even the morphology, therefore the shape of the given product or powder. So this is a nice little processing worksheet. It's available as a PDF that when we do training for people, we always review this, and I think it's something that's worth, wrapping your head around when you just start spray drying. So at the top of the chart are settings or parameters on the instrument that I can adjust or set. Down the side are things that we're looking to achieve. So if outlet temperature is important, or particle size, we're going to look at the chart and say the size of the arrow or the direction of the arrow is the impact. So let's say particle size is most important to us. This big arrow is my atomizing gas. So I would turn up the atomizing gas to bring down the particle size. Or as well, another large arrow is solid concentration. Okay? I want to go into these in a little bit greater detail. The sample freeze rate is in mils per minute coming from my nozzle. So the higher the freeze rate, the larger the wet droplet, the resulting the larger the bigger particle size. Also, if I have bigger wet droplets, I'm going to have greater evaporative cooling, therefore it's going to bring down my outlet temperature. And that will also, depending upon the other parameters, may give me higher moisture content, which makes sense. So conversely, lower feed, smaller droplets equals smaller particle size. I think one of the more important attributes is our outlet temperature. So as I said previously, I cannot set it. It's an effect of the other parameters. So we kind of have to wrap our head around this one a little bit more. There definitely is a misconception that if I'm using water, that I have to be at 100 degrees Celsius. That's absolutely not true in spray drying because I'm creating smaller droplets. So at the end of the day, I'm able to evaporate at lower outlet temperatures by adjusting my droplet size. And I think this is what really makes spray drying so attractive for temperature sensitive items. And the biggest impact on outlet temperature would be how hot my inlet temperature is. Atomizing gas flow, this is the gas coming to my nozzle, and this is depicted in liters of gas per hour. So once again, the higher the flow rate, the smaller the wet droplet is. I have quite a bit of leeway in my atomizing gas from an adjustment perspective. This will result in a smaller particle size and bring down the outlet temperature again. Once again, I have that cooling effect. So once again, the opposite is true. So the lower the flow rate, the larger the particle size is going to be. So we always get asked, what is the achievable particle size? So we say, at least on the B290, from a true solution, I am able to create particles in that 1 to 25 micron range. If I have a suspension, meaning I have a primary particle, it's a matter of particles being able to fit through the nozzle. Our standard nozzle is 0.7 millimeters or 700 microns. So we say about 1/10 of the size will fit through there. So about 70 microns would be the biggest primary particle. types of materials will impact my particle size as well as percent solid loading. Again, the size of the primary particles, if I have a slurry and I'm using a binder or carrier to agglomerate them, they're going to get larger. What process parameters that I'm employing, and a bigger nozzle doesn't necessarily mean a bigger particle. At the end of the day, it's more about ease of use. And sometimes if your droplets are too big, you just make a wet mess, and that's not what we want to accomplish either. Okay, so now we're going to move into the formulation end of things. So common excipients or carriers that are possible in spray drying are many, many choices, but they're typically within two classes of compounds, those that are soluble in water. And your excipient choice is really going to be, obviously, what market you're working in, what application. Is it something food related, where I might be using a maltodextrin or a dextrin? If I'm doing large molecules, I'm going to be using things like mannitol, trehalose, lactose, and even salt buffers. If I'm in the pharma landscape, I'm most likely going to be using synthetic biopolymers that I need to use organic solvent, not exclusively, but mostly. So these are the classes of compounds. Polymethyl methacrylate and their copolymers. There's many different kinds from many different vendors. I'm not listing any of the vendors, but just the chemicals. The PLAs, PLGAs, PVP, and their copolymers, hydroxypropyl methyl cellulose and its copolymers. And then on the material science side, we have some binders that will help with agglomeration. So we have a lot of different choices of what we're going to use to build our product. So I'm going to touch on encapsulation. When we do spray drying, we're going to use one formulation or single feed of our active ingredient and an encapsulant material to do what we call a matrix encapsulation. And what this means is that I want to have good content uniformity of my oil, flavor, active inside a solid particle that has a polymer or carrier on the outside. The point of this, quite frankly, is to just protect something, protect against degradation, oxidation, give it a better storage form, as well as taste masking. And I think more often I am picking excipients to give me controlled release of a given drug. Okay. So let's get into some of these examples that we were talking about. I'm going to start with food, flavors, and supplements kind of in a generic way. A lot of times, flavors or juices or natural products can be sticky, and they have what's called a high percent or high degree of bricks. And 1 brick is defined as 1 gram of sucrose for every 100 grams or mils of a solution. The higher degree of bricks that I have, the more sticky the solution is going to be. So I need an encapsulant. So you're going to need to pick an encapsulant that works best for your end product. Typically flavors would be maltodextrin and gum arabic. One thing I've learned over time that we really need proper mixing and good hydration, time for these excipients to fully dissolve. Okay. If I'm making an oil and water emulsion, that also is going to take time and other equipment to get a stable oil droplet. So that is part of the process. The other piece of it and this is a common theme through almost all of these applications. What is the ratio of my active oil to the encapsulant or carrier? And that's going to give me the best powder at the end of the day. If I'm using, maybe doing a supplement, do I need to worry about viability of it? The objective is I want to get good encapsulation efficiency and get something in a powdered form for later. Throughout these next slides, I'm going to have the literature references that I used for your ability to follow up later. So the first example that I'm using is a blackberry puree. This is the company that ironically came to our lab in Delaware to do a feasibility study. And through the feasibility study, we ended up writing a paper. I picked this example basically because it was overly sticky, and I thought it'd be a good example to show how the direct addition of a carrier basically went from being an unsuccessful spray drying run to a successful run. So the puree meat, we did 3 formulations that day. Pretty similar outlet temperature, resulting, similar inlet temperature, resulting outlet temperatures were similar as well. The first run had a very poor yield because the blackberry puree was very high in bricks. So we ended up adding mannitol in a 1-to-1 ratio, and then in a 1-to-2 ratio. And thereby increasing the amount of encapsulant, we therefore increased and made an unsuccessful spray drying day be a successful spray drying day. The purpose of this was also to retain polyphenolic compounds. They were going to make a supplement from this. Blackberries and many other fruit have nice antioxidants. So when they went back and did an analysis of it, using this thermoprotectant of mannitol, when they analyzed the 3 formulations, they were looking at the phenols before and what was the overall decrease in phenol content, and showing directly that through the encapsulation process, we were able to retain or maintain that intrinsic compound that they were looking to maintain. So my second example is going to be spray-dried solid dispersion. And as I said, this is essentially still the biggest area of spray drying for us. And essentially, the vast majority of the new APIs that are discovered are poorly aqueously soluble. So the idea is I want to add a biopolymer to them to help increase the bioavailability and the solubility. So I need to pick a given biopolymer based upon what they're looking to achieve and where they want this oral delivery to break down, whether it's in our stomach, whether it's in our intestine. I need to pick a solvent or a co-solvent system to get everything dissolved. I want to be mindful of the glass transition temperature of either the API itself or the biopolymer. Once again, this theme of proper ratio of our API to our polymer, the concentration or solids content within the pharma market, because things are poorly soluble. We tend to be in a low percent solid, typically less than 10%, could be as low as a couple of percent solids. And what other additives do I need? Do I need a surfactant? Do I need a dispersant? The first reference that I have here is done through the Waterford Institute in Ireland, and this is just a nice overall review of amorphous solid dispersions. It gives quite a few different references and talks about a lot of different biopolymers. So the third reference that I'm using is an example here doing a design of experiment. Now we looked at all of those parameters, which means I have quite a few different basically buttons to push and to understand what's going on. So they did a DOE where they have 16 runs and basically a 5 factorial. So they're using ibuprofen as a model drug because it has a very low TG and therefore highly crystalline. So through the act of spray drying, they want to convert this to an amorphous form. So they altered the solid concentration from 5 to 15%. They used a co-solvent system to get everything dissolved, both DCM and methanol. They also used a co-biopolymer system. So they were using the hydroxypropyl methylcellulose copolymer and a PVP vinyl acetate copolymer. One is an enteric coating, the other is going to help with solubility. And they also varied the ratio of the active to the polymer as well. Fairly mild spray drying conditions, hot enough to evaporate the DCM and the methanol, varied the feed rate, and kept the atomizing gas the same. From a results perspective, they analyzed for particle size, moisture, density, and the paper shows a lot of very nice graphs. But what I was more going to show you was the dissolution rate. So at the top, we have amount released, and at the bottom, time. We have 3 different examples that they picked. So the ibuprofen neat, being highly crystalline, is taking a longer time, up to 10 minutes or so, to fully get everything completely dissolved, or even up to 20 minutes. One example of the spray dried formulations was still crystalline, so it's taking a little bit longer, where the full amorphous formulation released in a quick amount of time. So therefore, I'm taking a very poorly soluble API and through spray drying, making it more bioavailable. And there are lots of literature examples of dispersion. Another area of importance is within the biotech, biotherapeutic area. Big molecules, including vaccines, things that are temperature sensitive, typically speaking. You need to understand that temperature sensitivity and keep your outlet temperature at or slightly below that. There can be some molecular weight concerns of bigger molecules. And typically speaking, we would say something under 50,000 kilodaltons makes more sense. If you have things that have higher molecular weight, you can combat that a little bit by lowering your concentration. Once again, I need a cryoprotectant or encapsulant, and we definitely have different choices when doing that. I think, again, it really depends on, what the end use 100% is for. Again, that reoccurring theme of ratio of my bioactive to my cryoprotectant and the use of surfactant. Here's an example using VSA as a model protein. Works really nice, and I think a lot of times if you have a new biopolymer or protein, you might want to use a model first before embarking upon a full study. Concentration is going to be important when it comes to morphology, and there's quite a few literature references that show if you use a surfactant, you really can change the morphology and the particle size. Here is BSA neat from an SEM. While here, you really went from being sort of granular to having very nice morphology, very spherical. So from a release perspective, this is going to work out better for us. One of the important attributes that spray drying can do for us. The last area I'm going to touch on is within the material science sector. Things like inorganic metals, nanomaterials, and ceramics. And this is also a very big area for spray drying. This is a little bit different in that I'm going to be using primary particles, and I'm going to manipulate these particles. I can agglomerate them. I can granulate them. However, most of the time I'm going to need a binder or a polymer, and typically they're in this area of polyvinyl alcohol or different pegs of different molecular weight. Graphene, I can actually fold it. Once again, I'm going to have higher loading in slurries versus true solutions, and I need to figure out the binder loading to my inorganic solid. I think we need to keep into account density and keeping these particles well suspended. A lot of them are very heavy from a dense perspective. Okay? Addition of a surfactant can really make a difference in, I'm going to say, the droplet formation. And most importantly, I've seen surfactants make a big difference with nanoparticles. They have really big surface areas, even though they're small, and they tend to clog. So a little bit of surfactant can make an unsuccessful run become a successful run. So let's get into a couple of examples here. This is an example of granulation, whereby which they were taking magnesium aluminum spinel and they were making transparent parts through casting. Very high loading, up to 40%, and essentially what they wanted to do was increase the bulk density and make these particles larger so that when they were casted, they basically came together and made a nice transparent film. So we went from basically, I think it said 200 nanometer particles and really making them much larger and therefore easier to manipulate and make a nice film. This was an example from India. The last example is an area that people have been working in for a while and still continue to do so, basically granulating of YSC or stabilized zirconia, so pretty typical ceramic. Once again, wanting to make bigger granules or agglomerates. This study showed taking the slurry in polyvinyl alcohol. PVA was at about 0.5% solids. And basically they were manipulating the formulation with different percent solids of a dispersant known as PAA, or I believe that the trade name is called Duramax. And this really changed the size of the granules. Once again, they were making fuel cells here. And as they sintered these to make a film, the size and the morphology really made a difference in the way that it performed from the tensile strength perspective and bulk density perspective. I just simply cut and paste the titles of all of these literature references here, trying to find some different references for this presentation today. There's 2 pages of that, and these are all in your handout so that you can look at them in greater depth. I realized that we just sort of touched on some things today, can't go through the paper fully. One thing I always do, and I'm sure you do as well, Google Scholar has many, many spray drying references. I certainly did a quick Google search and came up with 35,000 references. There are many patents. We ourselves, Buki's been doing spray drying for over 40 years. So we have many applications, short notes, and even a database that you can become a member of and download. And I guess I'd like to say, you don't have to reinvent the wheel. We host a decent amount of customers here at our site, and we spend a decent amount of time before somebody comes so that we're prepared to work with them. So at this point, I want to sort of wrap up my portion of the webinar before we get into questions. But essentially, I'll say spray drying is a way to quickly screen formulations on a laboratory scale. I think a lot of times doing a small DOE is a way to quickly come to a sweet spot of both the parameters and the formulation. Evaporative cooling allows us to do temperature-sensitive products. And through my formulation and parameters, I can define or develop particle size and morphology. One-step encapsulation to do case masking and content uniformity. And through spray dry and solid dispersions, really increase that bioavailability of poorly soluble drugs. A lot of times you can go directly to a tablet press, maybe with the addition of a few more products. Spray drying is attractive because it is a scalable process. And I'll just end by saying, my saying is, spray drying is twofold. And it takes formulation due diligence and process parameter control to really get to where you want to go. And now we have time for questions. Can results from a lab scale development directly be upscaled to large equipment? So we would say it's probably not 100% a one-for-one proposition, but most certainly can be scaled up. In fact, we have a nice best of Buki that talks about scaling up to the next size instrument, which is the mobile miner. But I think there's a very good correlation between lab scale and larger scale. Basically, that's the same question, in my opinion, is the scale-up challenges. I think the most important thing that you can do is spend enough time on the laboratory scale understanding the relationship of all of those parameters and what the impact is. One big difference for scale-up is particle size. So we can't fight physics. Okay, so we can get larger particles on a larger instrument. Okay.
Thank you.
We love Buki, but we have a lot of trouble scaling up because the production dryers do not hit similar temperatures. You have recommendations for production scale dryers that can hit lower temperatures. I'm probably not 100% an expert on scale-up instruments. I know that we have a marketing agreement with GEA, and I think the temperatures are similar from an inlet and an outlet. Sometimes the inlet temperature on production scale is higher. We really can't go much higher on a lab scale.
Okay, and here is another.
The flowability of the formulation, I'm not sure if we're talking about liquid or of powder itself. Flowability is going to be based upon, I think, percent solid. If it's flowability in the instrument, if maybe you need to decrease the percent solid to be able to reduce the viscosity to get something to and through the system. maybe we can talk a little bit more about this one offline. We have an intermediate spray drying unit in our pilot plant. Sometimes we notice loading of the product on the wall of the dryers. There's a specific reason for loading of the product. So within spray drying, we're never going to get 100% yield, and we're always going to have some sticking of product. If you're really getting lots of loading on the wall, I would say that something is too sticky, and you should increase the ratio of that excipient or carrier to combat that stickiness. Or maybe, conversely, your droplet size is too large in relationship to the heat, and you need to either turn up the heater or decrease your droplet size to get better drying. Yeah, so basically powders in themselves can be a little staticky. The B290 has basically a grounding group in there, and also the inside of our cyclones are coated with an anti-static discharge coating. I would say part of that has to do with particle size. A lot of times customers will have a place where they can control the powder afterwards, where they have a special hood where they're controlling the electrostatic discharge in the environment, which can help. On a lab scale, we sometimes use what's called an anti-stat gun. And I think a little bit this is intrinsic to powders, unfortunately. So yes, we do have an ultrasonic nozzle, and they're saying they have trouble with viscous solution. So the point of our ultrasonic nozzle was to more or less increase the particle size capability of the B290 because of the size of the drying cylinder. And there is a viscosity cutoff, and there's even a feed rate cutoff in order to not overburden the nozzle. So I would say, unfortunately, you would have to reduce the viscosity to give you the resulting powder quality that you're looking for. And I think it's just a cutoff with the kilohertz of the ultrasonic nozzle in itself. Aside from size, do you see good correlation to scale up in flowability and other properties? I think yes, we do. I think you're able to get the bulk density being similar, particle size, not necessarily so, but I believe other powder characteristics like good encapsulation and flowability or agglomeration should be accomplished, yeah. We often have particles that are half smooth, round, half round, but with wrinkled surface. We do not seem to influence the quality of the product, but are wondering what the wrinkling means and why particles look different within the same run. Probably has to do with the excipient that you're using, or even the active ingredient. Different excipients perform different ways. I would say maybe a little bit of surfactant might help with that droplet formation and then overall morphology, and maybe decreasing your percent solids, perhaps without having greater detail here. The batch size, we would say a gram is probably the smallest feasible. We have a high-performance cyclone with a small collection vessel that holds about 10 grams of product. Basically, you don't need a special nozzle for encapsulation, so just our standard 2-fluid nozzle will do just that. It's really about the formulation. I'd like to thank everybody for taking the time today to log in. I hope everybody picked up a little new knowledge of spray drying or something that you can use either in your current use or solidify your interest, use spray drying. And in summary, if you'd like to follow up, we can do so afterwards. I wish everyone a great day and be safe.